Chưa lên trên cái này này. Cái Google Meet này nó không thông báo như là cái Microsoft Teams, Microsoft Meet là nó mất tín hiệu nó thông báo, hay là Zoom mất tín hiệu nó cũng thông báo. Cái này nó chả thông báo gì. Okay.
In this experiment, a cell homogenate containing many different organelles is added to microtubules. Motor proteins are normally attached to the organelles. When ATP is added as a fuel for the motor proteins, some organelles bind microtubules and are moved along the tracks by their motors. Most kinesin motors move towards the plus end of microtubules. Dynein motors always move in the opposite direction. Both motors are used to transport organelles, and occasionally a single organelle, which must have both types of motor attached, can be seen to switch directions. The bidirectional traffic observed here is reminiscent of that in an intact cell.
So this movie
In this experiment,
was made by start to lysate the cell and take the cell extract and add it on to the like cover slip on the contained microtubule, and then ATP is added. When ATP is added, then the motor protein will work on microtubule, and as you can see,
a
many
cell homogenate
protein will go toward one end, while sometimes it go to the opposite end. That it's because they are bound by different motor protein, and there are two types of motor protein that can work on microtubules are kinesins and dyneins. There are only two types of dynein. One is tricytoplasmic dynein, another is axonomer dynein, which is cytoplasmic dyneins inside the cytoplasm, while the axonomer dynein are the dynein in the flagellar structure. Or then on the microtubules, on the dyneins, at least there's two type dynein, and then both of them move toward the minus end of microtubule. Meanwhile, the kinesin, there are many classes of kinesins, at least 14 classes of kinesins, and they not own, but most of them would move toward the plus end. Some of them move toward the minus end, and some even cannot move. So the movement of the organelle or transport vessel by motor protein can be explained by two models. One is the tug of war between motor protein. We have from the dynein and kinesins. If we have more dynein than kinesin, then this vesicle will be transported towards the minus end. But if we have more kinesin, then it will be transported toward the plus end. So it depends on the pulling force of kinesin and dynein, which one is would pull stronger, which one is stronger, then the vesicle will move toward the end of that uh, motor protein. Whether in the second model called as the correlation model of the motor activity, then it depends on whether dynein is activated or kinesin is activated, that the vesicle will move toward the minus end or toward the plus end. So if dynein is activated, then the vesicle or the organelle will be moved toward the minus end. When if the kinesin is activated, then the vesicle will be moved toward the plus end. So and also as you can see here, so every transport vesicle are bound by two types of the motor protein, and the interaction between those two types of motor protein will decide whether those vesicles can move towards the plus end or minus end. So this is the kinesin super family. Uh, we have here the kinesin 1, kinesin 2, to kinesin 14A, kinesin 14B. The name of kinesin is quite misleading because different type naming and classification. So probably quite difficult to follow all of this, but probably we only highlight some of it. So for start, the kinesin can be divided by three types, depend on the location, the position of the motor domain. Motor domain. So we have the end kinesin when the motor domain are located at the end terminal of the polypeptide. So we only care about the heavy chain, right? The heavy chain. So in here we own of N terminal or own of kinesin would move toward the plus end of microtubule. N kinesin move toward the plus end of microtubule, whereas the kinesin with the motor domain at the C terminal of polypeptide would move toward the minus end of the microtubule, which is the same direction as the dynein motor. And meanwhile, the M kinesin, where the motor domain located somewhere in the center of the cell, actually do not move on the microtubule. So this M kinesin cannot move on microtubule. Even though they have motor domain, they cannot move. So this belong to the kinesin 13. Kinesin 13. So we have KIP2A here, KIP2A. We have KIP2B, KIP2C, and KIP24. So all of these cannot move, and they actually involved in the degradation of microtubule. When they cannot move, they usually lead to dissociation of microtubule, tubulin from microtubule. And as you can see here, the classification of this one of this kind classification actually not following the phylogeny of those microtubule. So this is the phylogeny of those motor protein based on the motor domain. But the classification of terms are not following the phylogeny. This one probably a little bit difficult for us to explain to you, but this is how this motor domain can be shown to like in evolutionary evolutionary relationships. So we have like from your kinesin 1, KIP5B should be the oldest kinesin 5, kinesin 1, and then during evolution, it can lead to duplication and formation of the kinesin 5A and kinesin 5C. That is how it looks like. Usually, the grouping of this must follow that in one group, kinesin 1, all of the kinesin belong to one lineage, one evolutionary lineage, while kinesin 13, so we have KIP2A, KIP2B, and KIP2C are in one evolutionary lineage, while the KIP24 are different. It belong to another branch of evolution. So that is the problem with the classification of kinesin, and therefore the number of classes could be likely to be changed in the future. So the most studied kinesins are kinesin 1, kinesin 2, kinesin 5, and kinesin 13. So kinesin 1 and kinesin 2, they are they are dimer of the heavy chain. They are dimer of heavy chain. Kinesin 1 is a homodimer. Kinesin 2 is heterodimer. It also has some other light chain as well. For kinesin 5, actually it's a tetramer. it's a bipolar, which is somewhat similar to the bundle of myosin 2 when we studying actin. So the kinesin one is quite special. It can slide the actin from one another. Kinesin 13 they cannot move, and they lead to dissociation of tubulin from the microtubule. So this is how the kinesin one look like. It has the dimer of a heavy chain, it's a heavy chain, and it has two light chains. It had for each heavy chain, it has a head, a linker, and very long tail. The head would have the microtubule binding domain, and it also has the ATP binding domain, and it will also hydrolyze ATP to change the structure of the linker, which is equivalent to the net region of the myosin, and allow the kinesin to move on the microtubule. Even though they are different, kinesin and myosins, actually they originate from the same ancestor protein. So the movement of kinesins on microtubule a little bit different from myosins, in that it requires the coordination of two heavy chains for the movement. So if you still remember, then for myosin, each head will be moved independent of the other head. But in here, the movement of one head, it depends on the other. From here, we can start with the leading edge here. The leading edge as this stage, when it binds to ATP, it will bind to microtubule very tightly, and ATP binding to the leading edge head could lead to the changes in conformation of the linker region. And the linker, as you can see here, it's point toward the left side. When the ATP bind to it, it will turn to the right side. It turn from left to right. And when the linker of the leading head change from left to right, it will bring the trailing head at the back to the front. So the trailing head can move is because the trailing head is currently bind to ADP, and when it bind to ADP, it attaches loosely to the microtubule, and therefore it can change. And once it go to the trailing head, become the leading head, it will replace the ATP by the it replace ADP. It replace ADP by GTP, and then it will bind tightly to so in here it release the ATP, and it become tightly bind to microtubule. While the original leading head, now it become the trailing head, will hydrolyze ATP and release the phosphate. And when it bind to ADP, it bind loosely to the microtubule. So we have the alternative state of the strong binding of the leading head, when it is not bound. It's not bound by any nucleotide, or when it bind to ATP, and the loosely binding to ATP when it's bound to ADP. And the binding of ATP could lead to the changes in the conformation of the linker, or we can connect the net region if you want, so that it is easier to compare to myosin 2. So the binding of ATP to the leading head will have the movement of the trailing head, and slowly it's like that. So when the ATP bind to this one, the leading head here, the trailing head will be moved forward. This is the movie to illustrate
The
how
motor
it
protein
moves.
kinesin is a dimer with two identical motor heads. Each head consists of a catalytic core and a neck linker. In the cell, Kinesins pull organelles along microtubule tracks. The organelle attaches to the other end of the long coiled coil that holds the two motor heads together. The organelle is not shown here. In solution, both kinesin heads contain tightly bound ADP and move randomly, driven by Brownian motion. When one of the two kinesin heads encounters a microtubule, it binds tightly. Microtubule binding causes ADP to be released from the attached head. ATP then rapidly enters the empty nucleotide binding site. This nucleotide exchange triggers the neck linker to zipper onto the catalytic core. This action throws the second head forward and brings it near the next binding site on the microtubule. The attached trailing head hydrolyzes the ATP and releases phosphate. As the neck linker unzippers from the trailing head, the leading head exchanges its nucleotide and zippers its neck linker onto the catalytic core, and the cycle repeats. In this way, kinesin dimers move processively, step by step, along the microtubule.
Okay. So the kinesin at in normal state is also can be inactivated in the folic form, and it could be activated when it bind to the cargo. So for dynein, we have the cytoplasmic dynein would have only two heads, while the ciliary dynein or the axonal dynein would have three heads. Yeah, it had three heads, and sometimes they do have the dynein or they had only one head, a single head in the ciliary. So dynein is a very large protein. It's a very large protein. This is one heavy chain, and it has multiple light chains. It also works at the monomer, but it contains two heavy chains. So in here we have the microtubule binding site. It has a stalk, and it has 6 domains of ATPase. The orientation organization is could be easier to follow here. So we have the stem with this one, and the linker, and this part linker, and so we had 4 domain 1, 2, 3, 4, and then it has stalk. The stalk will fold on itself, and then domain 5 and 6. Among these 6 ATP domains, it's only known that the ATP domain 1 is still has the ATP activities, and it can hydrolyze the ATP. So the hydrolyzation of ATP by domain 1 is contribute to the movement of dynein, and the others are not involved in the hydrolysis of ATP. So when the ATP is hydrolyzed, then the configuration of linker is changed. So as we can see, it's in a curve form, and then it changed to the straight form. When the linker change, you can imagine that this ATP domain here, it will rotate, so make the domain rotate. And with the rotation of the ATPase domains, the stalk will move from left to right, and make the make the dynein move on microtubule. Or if the dynein are attached stationary, then we will have the microtubule will be slide. So when the ATP hydrolyzed, then the stalk will change the location because of rotation of the ATP domain. The stalk will change the position and it slide the ATP around 8 nanometer. So the movement of dynein and kinesin is only around 8 nanometer. It's shorter, much shorter compared to microtubule. Here's two different forms, two different states of the dynein heavy chains. So we have a stock here where it's bind to microtubule. We have a linker region in here. Region in now is in the curve form, but later when it comes in straight form, then it allow the changes in the relative location. Sorry, this one is the stock. The stock is a relative location of the stock compared to the ATP domains, and the changes in location of stock could lead to sliding of microtubule. Here's how it works. We have a ATP bind to the ATP domain 1 and the hydrolyzation of it changes the linker. Release the phosphate and return to the original state. Release of the calcium ADP. It can be divided to multiple states. It's a little bit complicated, so okay. Probably it better to watch this movie. This is how it works. The movement of dining on microgrid is not always regular. Sometimes it change direction. It's people compare it to like drunken man walking. The dining actually cannot work alone. Dining has to work when it's in the combination with the dynactin, a large complex containing actin and actin related protein 1 filament bounded by the Cap-G and polytropomodulin to stabilize short stretch of actin-like filament. And there's the dynactin protein would link the dynactin complex to the dynein. And beside this, they also have a P150 glues. These proteins function is to keep like tether the dynein to the microtubule and keeping the dynein all the way attached to microtubule during the movement. And the two head of dynein when it's hydrolyzed, the ATP will change the conformation of the linker and allow the microtubule to move. So the dining only way work at a very large complex, it's huge complex. Therefore, people it's much more difficult to study compared to dining. The microtubule dynein and kinesin involved in the formation of microtubule spindles as well as the separation of chromosome during cell divisions. So microtubule from the mitotic spindle can be divided in three types of microtubules. One is archer microtubule, the microtubule which originated from the centrosome and attached, usually contact with the cell membrane. And the archer microtubule from both ends of the mitotic spindle will make the contact with the cell membrane somewhere here, and it will mark the plane where it will mark the equatorial plane. It's also it's marked where the cytokinesis will happen, where the cell will be divided into two smaller daughter cells. So the astra microtubule will beside the cell division plane. Then we have the kinetochore microtubule, which are microtubule attached to chromosome, the kinetochore on the chromosome, and these are the microtubule directly involved in separation of the chromosome. And then we also have an interpolar microtubule, which microtubule originate from centrosome and interact with each other in the center of spindle. These red black dots are actually myosin 5, the bipolar kinesin that can slide the microtubule. Probably we have action is in here. So kinesin 5 can slide the microtubule, and therefore when it walks toward this blood end and this blood end, it will push the microtubule further apart, and it pushes the two centrosomes further away from each other. So that is, and then together with the pushing of kinesin 5, the microtubule keep growing on the plus end, and therefore during the anaphase, we can see the elongation of mitotic spindle, as well as the separation of the two sets of the microtubule, two sets of chromosome attached to the kinetochore microtubule. So this is the kinetochore microtubule, where the kinesin 4 or kinesin 10 would involve in the transport of microtubule chromosomes toward the plus end, where it will orient in the metaphase plane. It will be arranged in the metaphase plane. And later stage, the movement of the microtubule here is also involved in dynein, which is not drawing here. The dynein will attach to the plus end, and when it moves the minus end, it will pull the microtubule or pull the centrosome close to the cells toward the cell membrane, and also involved in elongation of the spindle and enlargement of the spindle using cell division. And we also had the kinesin 14. Kinesin 14 is actually only one set, one class of kinesin moving toward the minus end of microtubule, and also it can involve in the dynamic of mitotic spindle. So I don't have the figure here. So we have the microtubule, the chromosome attached to plus end of microtubule, and then we have the dynein attached to the kinetochore, and then it keep moving toward the minus end, while the plus end it keep shortening, and therefore it's allow the chromosome to be transported close to the centrosome, and later they will form a new nuclei. So inside the cells, in animal cells, the intercellular transports are actually mainly done by kinesin and myosins. So we have the endoplasmic reticulum usually stretched toward the peripheral of the cells by kinesins, while the Golgi apparatus are focusing to the center of the cells toward the minus end by dynein. The mitochondria are transported to the location of cell that require energy by both kinesin and dynein, and pigment granule also be transported by both type dyneins and kinesins. And the endosome it depend on the late endosome, and they could be transport from the outer region of the cell to the center region of cells by dynein. And many other organelles on their own in animals. Any organelles are transported by motor protein on microtubule. So this is how the ER and the Golgi apparatus look like. We have the ER network, which are very in the same location at the microtubule, and we have the Golgi apparatus concentrated around the centrosome, where the microtubule are most dense. Melanosome from depend on the signals from when the cyclic AMP is reduced, then the kinesin will be activated, and therefore the pigment granule will be focused toward the center of the cells, and the cell will look like brighter, have a brighter color, because less light will be absorbed by the cells. When the cyclic AMP concentration is increased, then kinesin is activated, and then kinesin will move the pigment granule toward throughout the cell body, and make the cell become darker, because it will absorb more light. Microtubule are very important for establishment of the cell polarity, because it involved in transport of many determinant. As in here, the microtubule can be grow from the centrosome, but depend on some region of the cells, where the certain factor like microtubule stabilizing protein, it can stabilize microtubule and leading to formation of the parallel bundle of microtubule, and then this bundle of microtubule will serve as a track for movement of many determinant toward the one location of the cell to establish cell polarity. And I think probably have you studied developmental biology? You have not, right? So later you will study developmental biology, in that the establishment of the embryo axis, like the anterior, posterior, dorsal, ventral axis, are dependent heavily on microtubule, on determinant that are transported by microtubule. In here, for example, shown by illustrated by the organization of microtubule during the oocyte development, Drosophila oocyte development. At earlier stage, the nurse cells will synthesize many RNA and protein, and then those RNA and protein will be transported to egg cells, and the microtubule organized with the plus end in the nurse cell and the minus end in the egg cells allow the movement transport of glycan protein by dynein toward the minus end, and it establishes the posterior of the oocyte. And later, the signal from the posterior follicle cells will lead to the changes in organization of microtubule with the blood end at the center, and now the nucleus will be start at the center here, will be pushed towards one side of the oocyte, and this side will become the dorsal and also the anterior side of the embryo. It will become a dorsal side. This is the dorsal side. And the movement of the nucleus here is dependent on the microtubule. People had already observed the pushing of the nucleus by microtubule toward the future dorsal side. And the changes in organization of microtubule in later state lead to accumulation of OSCA RNA at the posterior end, which lead to the formation of the belly part, the abdomen of the larva, where the bicoid will establish the head region of the larva, and this lead to the formation of the axis of a new body. We have anterior marked by bicoid, posterior marked by OSCA RNA, and dorsal marked by the Gergen protein. And when the anterior and posterior axis and the dorsal ventral axis is established, we also have the left and right axis established in Drosophila embryo. For multi-organ, for other cell like neurons, or neuronal cells, in neurons, then we have a lot of microtubule that form the track for transported of RNA and proteins toward the synapses. We have axon and we have a dendrite here. So all the RNA are synthesized inside the nucleus and transported by microtubule to certain location of the cell and then translated into protein involved in the function of the neuronal cells. Then we in those regions we usually have microtubule which are oriented with the plus end towards the periphery of the cell, when the minus end toward the center of the cells, and it serves as a track for the transport of the outward transport by kinesin and inward transport by dynein. Or sometimes it's called as anterograde transport by kinesin and retrograde by dynein. And the two last structure made by microtubule can be observed on the cell surface. Cilia, we have multiple cilia. Cilia are usually short, and one cell have many cilia compared to other structural cone at flagellum. So in here is the epithelial cell in the respiratory tract of the pig. And we have some cell with a smooth cell. We do not have the cilia. This cell will secrete the mucus due to trap dust and bacterial viruses. Whether we have some other cell with a lot of cilia to move this mucus out respiratory tracts and to keep the respiratory in a sterile condition. And actually cannot be sterile, but actually in the good condition. So here is how it works. This is smooth cell. It will secrete mucus, and these are the cell with cilia beating in synchrony so that it allow the movement of the mucus out of the respiratory tract. And with this movement, all the dust bacteria will be clear from the respiratory tracts and maintains our good healthy respiratory system. The other structure that control made by microtubule are flagellum. So structure of flagellum and cilium are the same. They have the same structure, and it depends on the region of the flagellum or cilium. We have at the basal body could have 9 sets of 3 microtubule. These 3 microtubule will name as A, B, and C, whereas the A tubules contain 13 protofilament, whereas the C and B tubule contain only 10 protofilament. And the A and B tubes will be served as template to form A and B tube in the axonyms or in the transitional zone, where we have in the basal body we have the center region. All of this microtubule attached to by the other protein to the center tube. Whereas in the transitional zone, there's none structure at the center, and in the axonym regions we have the two pairs, a pair of microtubule at the center, and we have 9 pair of microtubule in the outer region. In some here we have some short part here are dynein. These are dynein dynein hand. Probably it's easier to look at the illustration here. We have these are dynein hands on a microtubule A attached to a microtubule A, and it will interact with the microtubule B in the next duplet. And these microtubule duplets are attached to each other by protein called nextings. So nexting you constrain the movement of this microtubule, and they can microtubule here can also interact with the other protein to form the stable structure of cilium or flagellum. The ciliar dynein can had 3 heads and sometimes had 2 heads. It's attached to the A tube, and then it interact with the B tube. When the dynein moves on microtubule, it will slide the microtubule. However, inside the cilium or flagellum, these two microtubule, these two doublets of microtubule are fixed together by nexting. Then they cannot move it, they cannot slide. Therefore, when dyneins move on the microtubule, it will bend the microtubule duplet. It bends microtubule, and therefore it bends cilium and it bends flagellum. And this bending of the cilium and flagellum lead to the power strokes allow the movement of the cell forward or allow the movement of muscles outside of the cells. And later it will the combination of the dynein along the cilium or flagellum, it will allow the flagellum and cilium return to original state in the recovery stroke. And cilia as well as flagellum is very important for cell movement. As you can see here, it's a parasimium with a lot of cilia on its surface, and the movement of this or the beating of this cilia allow the flagellum parasimium to move around. And the beating of flagellum also have to propel the sperms forwards when they move to fertilize the egg. So flagellum, in terms of structure, flagellum and cilia have the same structure, has the same organization of microtubule, but the only difference that one cell may have many cilia, but they usually only have one or just a few flagella, and the flagella are also much longer than cilia.
In this video, we'll be looking at sperm specimens with varying motility scores utilizing the microscopes in our fertility
We can observe the movement of sperm under the microscope,
laboratory.
and they use for fertility test.
Sperm motility is pretty good. Most sperm motility scores.
So this sperm, and when the flagellum bands, they go move.
Represent the percentage of sperm that are actively swimming in a forward direction. Low sperm motility with a good count and a good motility. Here it is at higher magnification, where we can get a better idea of the morphology or shape of the sperm cells. This specimen.
Okay. And there's one special type of cilia called primary cilia. Actually, this is immobile cilia. Cilia, it cannot move. And this primary cilium is observed in almost all types of the cells. So multicilial is only observed in some type of cells, but primary cilia is observed in all types of the cells. It cannot move. Okay, so remember that primary cilia cannot move, and it's formed from the mother centrioles. It's from the mother centriole. It has the most basal body is the mother centriole, and it's only observed during the interphase by the cell division. Then the primary cilium will disappear, because the centriole will have to contribute to formation of mitotic spindle. But primary cilia is very important for cell sensing. There's a lot of receptor on it to have allow the cell to sense the environment, and also involved in many other processes. The last type of the cytoskeletal filament is the intermediate filament. So intermediate filament can be observed on like epithelial cell, skin cell, where you can see that it looks like the intermediate filament coming from one cell and go through the cell membrane to the other cells. So these are the desmosomes, where the cells are connected tightly to each other by intermediate filament. So intermediate filament is different from actin filament or microtubule in that the monomer are actually filamentous monomer instead of globular monomer of the actin or alpha and beta tubulin. So intermediate filament is formed by filamentous monomer, and this monomer will combine together to form a dimer with the same orientation of the N terminal and C terminal. And there's a multiple type of the monomer inside the cell. They can combine to form a homodimer or form heterodimer, and therefore the intermediate filament is very diverse inside the cells. And actually, the intermediate filament is can be specific to some type of the cells. For the keratin can only observed in skin, and it can be used to track the cell movements. For example, in cancer cell, when cancer cell migrate to some other region, if we observe it has expressed the keratin, we know that those cells may originated from the epithelial cells or skin cancer, for example. And the dimer can combine together to form a tetramer in the opposite direction. They orient in the opposite direction. The two N terminal will combine with the two C terminal at one end, and also have this form a stacker structure where it has a gap. It has a gap at the end, and this gap allow them to combine together to form like protofilament. So the formation of intermediate filaments had not very well known. So some models suggest that 8 tetramer can combine together to form a bundle, and this bundle of 8 tetramer will interact with each other by reach a stacker region to form a large filament. And some other models suggest that each tetramer will interact at the end here to form a protofilament, and 8 protofilament will form one intermediate filament. So we actually don't know which one is correct, because the intermediate filament is start to be like to get inserted in recent years, and we don't know much about the intermediate filament. In terms of classification, there are several system of classifications. So they can be divided into two parts. One is a nuclear and intermediate filament, where we have nuclear lamine, which form the lamina inside under the nuclear envelope, and then we have the cytoplasmic intermediate. So nuclear intermediate filaments is observed in all of the in all cells, in all the eukaryotic cells, but the cytoplasmic intermediate filament is only observed in animal cells, in animal cell, and only in some type animal cell. For example, the keratin filament is only observed in epithelial cells. In some system, they separate the keratin into small classes, keratin 1 and keratin 2. And pimentin we have in connective tissues, and neurofilaments only observed in nerve cell. So for plant and fungi, they do not have cytoplasmic intermediate filament. The keratin function in human, there's around 50 genes in human cells, in human genomes, and is expressed specifically in epithelial cells, and it contributes to strength of the skin. If the genes encoding for keratin is mutated, then the epithelial cell is quite easily to be destroyed, and it leads to the disease. So that means it's a type of pimentin is part of the structure muscle in its smooth muscle as well as the skeletal muscle. Neurofilament, especially in the neurons cells, and it strengthens the axon, it strengthens the dendrites. This is cross section. We have a lot of neurofilament and some of the microtubule here. So disruption of neurofilament also leading to neural disease, because the dendrites and axon cannot grow longer. The nuclear lamina could observe in all of eukaryotic cells is formed under the inner nuclear membrane, and it interact with the chromatin to lead to the formation of heterochromatin in the region interaction with the nuclear lamina. It forms a regular network, and proliferation of lamine in the prophase at the end of prophase lead to the assembly of nuclear envelope. In humans, there are 3 genes encoding for lamines, lamine A, lamine B1, lamine B2, but there are 4 different proteins. In that, lamine A, B1 and B2 are expressed in all the cells, but lamine A and C are regulated. The expression of it will be controlled by cell development and specification. The own 3 types of skeleton fibers are interact with each other by multiple protein. For example, plectins can cross-link microtubule to intermediate filaments, and protein that containing Cas and Sun domain can cross-link intermediate filament inside the nucleus with the outer structure like actin, microtubules, and intermediate filament in the cytoplasm. And all the component of cytoskeleton actually interact with each other to maintain the cell shapes, to support the cells as well as involved in the cells movement. Okay, probably we stop here. Do you have any question? Hello. Are you still there?
Yes.
Các bạn có hỏi gì không ạ?
Dạ đến giờ thì chưa ạ. Em nghĩ là bọn em sẽ về đọc lại một lần nữa, xong rồi có gì bọn em lại ngồi sau ạ.
Rồi, được rồi. Ok, các bạn có thể đọc thêm sách nữa, tại vì cái slide thì nó không có nhiều lắm đâu. Nếu mà không có hỏi gì thì chắc là chúng ta nghỉ đây.